can't believe this thing's not working. Been trying this all day to run the samples over and over again. Oh hey, I'm not getting my results here.
What's the problem?
Well, I took patient's DNA from their blood sample and put it on the machine, but it's not reading it. I don't know, maybe we don't have enough DNA in here.
It doesn't look like it.
Oh, I don't know how you can tell that, but we need to make some more copies of this.
Have you tried the copy machine?
Alright, let's go try it out.
Alright, let's go.
Alright, here we go. I'm going to try to make some copies of this DNA. Let's see how this thing works. Oh, this is not going to work. This doesn't even fit in here. How else can we make copies of this DNA?
Have you thought about PCR?
Oh, PCR. Why didn't I think of that before? Yeah, we were just learning about that, the polymerase chain reaction. That makes a ton of copies of DNA. Alright, let's go set that up. This copy machine is not going to help us out. Alright, so we got all my ingredients here for the PCR. Let's give you guys a quick look at that. See if we can get a good shot. We have distilled water, which is most of the mix. Some buffer, MgCl2, our free nucleotides so that they can bind to the template DNA. We have our forward primers and our reverse primers so that we can target a specific sequence of DNA. Our genomic DNA, which we could get from various sources like hair or blood. And our Taq polymerase, which is going to actually add the nucleotides. So now what we got to do is use our pipettes and add these different ingredients together and run the PCR. So I'll show you real quick how to use the pipette. So first you set it up to a specific amount, and there's 3 different pipettes we use. So this is now set for 6 microliters. You go get a fresh tip every single time. And when you first go in, you push the plunger down to the first stopper, put it into the tube, drop your sample, and when you transfer it to your new vial, you push down to the second stopper. And basically what you have to do is set each pipette and put in the precise amounts of each ingredient into your PCR cocktail.
Seems so simple.
Not that hard. Alright, so now what we're going to do is take our PCR mix and put it into our PCR tubes. The PCR thermocycler we're going to use fits these size tubes, so we have to transfer that up. And I set my pipette here, 12 microliters. Alright. Alright, and once I have all of the liquid aliquoted out, we're ready to run our PCR. So we're going to go over to the thermocycler and show you guys how to set it up. Alright, so here we have our 4 thermocyclers, which can all do PCR. We just have this last one open, so I'll show you guys how to set it up. And the key here is just to set the correct temperatures. And temperature is what's going to make the PCR happen. So for our first step, we have at 95 degrees. We'll set that for 9 minutes. And basically what this first step does is denatures the DNA, so it separates the DNA strands. And then we go through this cycle of 3. So it starts off with 95 degrees again for 30 seconds, which is again going to take that double-stranded DNA apart so that the primers can attach. That's our next step, is 30 seconds at 55 degrees. And this is where our primers anneal, and that's going to direct our next step, which is at 72 degrees, where our Taq DNA polymerase is going to add the nucleotides to the template DNA. And as you can see, this is run 34 times. So every cycle you should get double the amount of DNA until you have millions of strands of a specific sequence of DNA. Alright, so now we have our PCR set up. So to take our 8 well PCR tubes, put them in the PCR machine, our thermocycler, close it up, and hit start. And the process is going to take a couple hours, depending on how many cycles we run it. It could take longer or shorter. So if you want more amplification, we could do 35 cycles instead of 30, and I'll just add on a few minutes or an hour to the process. Alright, so our PCR is finished, and we take it out of our PCR thermocycler, and we want to try to see if there's been amplification. I'm going to put on my glasses, which I got from Mr. Coy, see if I can get a better look, but can't really tell. Ryan, can you zoom in there, see if there's amplification or not?
Can't see.
Can't see. Alright, well, basically it's impossible to see with the naked eye or with these ridiculously strong glasses. So we're going to have to run a gel, and that will separate our strands by size, and we'll be able to tell if our PCR worked or not. Alright, so now I'm going to show you guys how to make a gel. The gel is going to basically separate the DNA strands by size so we can see what we got. So we have to construct our gel. The first thing you need is some buffer. We're going to use 210 microliters, which means I'm going to fill this graduated cylinder up twice and then add 10 more microliters. It doesn't have to be very exact, but actually the more buffer you put in will make the gel a little bit less dense, so the DNA will travel faster. If you have less buffer, it'll travel a little bit more slowly. Alright, so now we have our buffer in there. We're going to go put in the agarose. So here we have our agarose, which is going to make it solidify. And we have to use our balance to make sure we're accurate. So we put our weigh boat on there and tare it so it goes down to 0. And we need to put in 3.15 grams of our agarose.
That's pretty good.
That's enough. We add that directly into our beaker full of the buffer. Swirl it around. Now we actually have to microwave this so that the agarose and the buffer come together and it all gets dissolved. Let's go to the microwave. A trick I learned here was to put a few Kim wipes in the top so that we don't lose so much of the buffer so it doesn't evaporate away. We come to our microwave and we set it for about 4 or 5 minutes, and we'll come back when it's all done. Alright, now you can see all the agarose has been incorporated into the buffer, so we have a clear solution here. And what we have to do now is just let this cool. So let this cool for about 5 minutes, and then we can put in our ethidium bromide. Ethidium bromide is pretty toxic. You never want to get it on your skin, but what it's going to do is actually attach to the DNA and tag it, so when we put it under the UV light, we can see where our bands are. So right now, we'll let it cool, and then we'll put some ethidium bromide in there. Alright, so now we're going to put the ethidium bromide in there, which again, don't get this on your skin because it's cancerous. So we put this into our solution. And that's pretty much it. Now we're going to go take this over to our gel mold. Alright, so here we are. We have our gel mold right here. Find these in various sizes depending on what size gel you want to make. And then we have these combs which are going to create the wells where we're going to put our PCR sample. So put your combs in. And then we take our agarose solution. Just pour it in. And we have to wait about 10 minutes for this to solidify, and our gel's ready to go. So we'll come back in 10 minutes. Alright guys, so this is about 10 minutes later. Check the gel out. It's pretty hard. You just pull the combs out. And this will give us our wells for the PCR cocktail to go in. Just take your gel, put it in the gel box. We previously filled this box up with a buffer solution. It's going to conduct the electricity we need to move the DNA. Alright, so now we're ready to load the gel. And I've put about 2 microliters of dye in the PCR tube. And this is going to make our solution, our mixture, sink so that it sits in the well and doesn't float up. And then we have our PCR, which we just ran. So I'm going to use this 8-channel pipette to make it easier, so we can do 8 at once, or you can do 1 at a time. But I prefer this because it takes less time. So you kind of have to slam the pipette in to get the tips to stick. We're going to take out 5 microliters of the PCR cocktail. Put that into the dye. Pipette up and down. Then we're going to load this up. It's important when you load it to make sure the pipette tips are actually in the wells. You simply dispense and pull it out. So now we have 8 channels loaded. I didn't really want all that dye to come out, but most of the PCR cocktail got into the well, so that should be fine. And the one other thing we need to add is a ladder, so that when this DNA travels through the gel, we have to have something to compare it to. So we have our 1 kb plus ladder, which means it's ideal for something between 100 and 1,000 base pairs, and we're looking for 350 base pairs, so this is good for us. This is just a single channel pipette. We'll use that to load our ladder in. Alright. And when you get close to the well, you usually use your finger to guide it in. So it has to be pretty precise. That looks pretty good. Alright guys, so now we have to turn on our gel electrophoresis box. And we do that by just hooking up electricity. So we want to make sure that it goes from negative to positive, so that the negative DNA is attracted to the positive cathode. We plug the other end into our device that's going to give us our voltage. Turn it on. Alright, so our gel's been running for actually 45 minutes, and I'm going to turn it off. It's done. Disconnect the electricity, and you can see right here that the dye moved, so we know that movement happened. Now we have to take it to our gel reader and see if the DNA amplified. Alright. We have our gel, and we need to load it into our gel documentation system, which is a camera hooked up to this box, and the box can emit white light or UV light, and what we want is the UV light, which is going to illuminate our DNA, which has been tagged with the ethidium bromide, so that we can hopefully see the PCR amplified. Check out our computer screen. This is our UV light, and you can tell right here that actually our PCR works perfectly. I was expecting about 349 base pairs, and our ladder goes, each band of the ladder is about 100. It is 100, so we go 100, 200, 300. Our band is between 300 and 400 base pairs, so our PCR was successful. Alright, and that's basically it. We ran our PCR, you check your amplification by running a gel, and you're good to go. Alright, so I took that PCR that we ran successfully, checked it on our gel, we popped it back in the CE machine, and this thing takes about 4 or 5 hours. I didn't want you guys to see that, but what I do want you to see is that our sequences work. So after we did PCR, we got our amplicon, and now we're ready to analyze our results. So we can find any number of things, but what we're looking for here are variation in these patients' DNA. All right? I want to thank you for joining me with our PCR and gel electrophoresis lab here at the Cutting Labs at Johns Hopkins Med School.